Friends, welcome to Biology by DG. Today in this video, we'll discuss about the structure of the cell membrane. So the cell has a membrane that is known as cell membrane or plasma membrane or plasma lemma. That its presence was first discovered by Swan. Swan in 1838 discovered the presence of a membrane in the cell, and this the cell membrane, it is the outermost layer in case of animal cells, while in case of plant cells it is just inner to the cell wall. The term cell membrane, it was given by Nageli and Kramer. Nageli and Kramer, 1855, they have coined the term that is cell membrane. And the cell membrane is also known as plasma lemma, and the term was given by Plow. Plow, he gave the, that is in 1931, and he gave the name plasma lemma. So you may call it as plasma lemma or cell membrane or even plasma membrane. Now this membrane is present in all types of cells, whether they are prokaryotes or they are eukaryotes. Even in eukaryotes, there are different organelles like mitochondria, plastids, endoplasmic reticulum, Golgi body, as well as nucleus is there. That means all these membranes are almost similar in structure. That is why the membranes are also known as biomembranes. Biomembranes means all the membranes that are present in the cell. Now in this, today in this video we will discuss about the chemical composition of this cell membrane. Chemical composition. So the chemical composition of the cell membrane was studied taking the RBC, and it was found that the cell membrane consists of or it is a lipoprotein. That means it consists of lipid and protein, and these lipid and proteins, they are present in the ratio of 40:60. That means lipids are 40% and proteins are 50%. But particularly in case of RBC, this is the general rule. But in case of RBC, this ratio is usually 40:52. That is 40 lipids and 52 proteins. Now, though they are mainly consist of lipids and protein, the ratio of 40:60, or in case of RBC, that is 40:52, it is usually variable because it depends upon the type of function the cell membrane carries. Suppose the cell membrane, the nuclear membrane, or you take any membrane, endoplasmic reticulum, plastid, Golgi body, since the different membranes have different functions, that is why this ratio is not constant, it is variable. Even if you take the case of a mitochondria, in mitochondria there are two membranes, outer membrane and inner membrane. In inner membrane there are more proteins than the outer membrane. So it is always variable, but in average we take this, that is 40:60, this is the average. Now, this besides these lipids and proteins, it also consists of some amount of carbohydrates. That is ultimately the cell membrane consists of lipids, proteins and carbohydrates. Carbohydrates usually they range from 1 to 5% only, where the major are lipids and proteins. Now we will discuss one by one what are the different types of lipids, proteins and carbohydrates that constitute the cell membrane. Now first we will discuss the lipids. Usually the lipids which are present in the cell membrane, they are of different types, or we can tell that is diverse types of lipids are there. Diverse type, that means different types of lipids are there, but whatever type may be there, most of them are amphipathic. This is very important. They are amphipathic. Now, what is the meaning of this amphipathic? That means the lipids have one hydrophilic head, polar head. So we can draw like this. That means this is the head, which is hydrophilic, and these are the two tails, which are usually hydrophobic. So when both polar and non-polar or hydrophilic and hydrophobic are there, they are known as amphipathic. This is very important from examination point of view, amphipathic. And they are present, most of them, the lipids there, this is amphipathic. Now these lipids, since already I have told you that are diverse types, they are again of different types. We can tell that there may be phosphoglycerides, there may be sphingolipids, and the third there may be cholesterols. These are the different types of lipids that are present in the cell membrane, but all of them are usually amphipathic. Now let us discuss one by one. Phosphoglycerides. Most of the phospholipids that are present in the cell membrane, they are phosphoglycerides. So, it will one of the phosphoglyceride, you can see this is the structure of one phosphoglyceride having one polar head. This is a polar head, and these are two non-polar tails. That means here one important thing is, usually in a fatty acid, usually three fatty acids are present in a normal one. But here one of the fatty acid is substituted by phosphoric acid. That is why the name phospholipid. That means out of three, one is substituted by phosphoric acid. That is why only two fatty acids are there. So here one interesting thing is, the fatty acids which are stored in the cell, they have three fatty acids. That means they are triglycerides. That means in this form they are stored in the cell. But those which take part in the cell structure, that is in the form of phospholipids, since one is substituted by phosphoric acid, they are usually diglycerides. This is unique to them. And the two fatty acids which are present, these two fatty acids both may be saturated, both may be unsaturated, both may be polyunsaturated, or one may be saturated, the other may be unsaturated. But in most of the phospholipids, one is saturated, the other is unsaturated. That is, one is saturated and the other is unsaturated. This is a normal case. So this is about the different types of phosphoglycerides that is present in the cell membrane. Now comes the second one, the sphingolipids. Now sphingolipids are usually very rare in the cell membranes. So the sphingolipids, these are usually very rare, and they are usually derivatives of that is amino alcohol, derivatives of amino alcohol, that is sphingosine. So sphingosine is one amino alcohol. So that is derivative, they are known as sphingolipids. So what are the different types of derivatives? Since it is a derivative amino alcohol, so naturally there will be one alcohol group. Now if that alcohol group is substituted is substituted by, the first one is phosphoryl choline. Then the type is known as sphingomyelin. That means the phospholipid is known as sphingomyelin if the alcohol group is replaced by phosphoryl choline. Now, if it is replaced by carbohydrate, then it is known as a glycolipid. If it is replaced by sugar, then it is known as cerebrosides. And if it is replaced by a cluster of sugars, then it is known as a ganglioside. That means the different types of sphingolipids that are present in the cell membrane, they may be sphingomyelin, glycolipid, cerebroside or ganglioside. But in all these cases, the glycerol is present. But in this case, glycerol is absent. Glycerol backbone absent. So these are the different types of sphingolipids. Then the next one is cholesterol. Now usually the amount of cholesterols that constitute about 20% of all the phospholipids. The cholesterols, they constitute about 20% of lipids. And particularly in case of animal cells, they constitute about 50% of lipids in animal cells. But you see, in case of animal cells, 50% of the lipids constitute only cholesterol. But this cholesterol is absent in case of plant cells and bacterial cells. Absent in plants and bacteria. But in case of bacteria, there is a substitute to this cholesterol. They are usually known as hopanoids. In case of bacteria, the substitute is known as hopanoids, or simply you can tell hopanoid. Now, what is the function of this cholesterol? Usually the cholesterol provides a rigidity, strength to the cell membrane. The main function is it provides a rigidity to that of the, this is the important function of the cholesterol. So, though if there will be, that means cholesterol is very important for the structure of the cell membrane. But if this cholesterol will be more in blood, then it will lead to, that is hypertension. So these are the different types of lipids. Now, since these lipids constitute about 40% of the lipids, what is their main function in the cell membrane? That part we now will discuss. What is the main function? So the main function of the lipids, or you can tell the importance or nature of lipids. Now, one of the importance is they form extensive network in the cell membrane. That is one important. The second one is provides a fluidity to the cell membrane. Now this provides fluidity to cell membrane. This is very important, very important point, because it is because of this fluidity all the functions of the cell membrane are carried out. So what are the different functions? Due to this fluidity, the different functions are, that is cell growth, then cell division, secretion, then endocytosis, then intercellular junctions. These are the important functions of the cell membrane because it has a fluid structure. It provides fluidity. As a result, the cell grows in size, the cell divides, the secretion, excretion, and endocytosis, that is the formation of phagosome, pinosome, and intercellular junctions. These are the important functions because it is fluid in nature. That is one of the very important functions. And in addition to this, another function is it has capacity of self-assembly. Self-assembly. That means if a cell membrane is disrupted, then again it can reassemble. So these are the important nature or functions of the lipids. Now let us come to the next, that is the proteins.